So today we're going to talk about beta oxidation of fatty acids. And before we talk about that, I'd like to talk about why we use fatty acids. So fatty acids contain 9 kilocalories per gram consumed, which is more than twice as much as carbohydrates and protein, which only have 4 kilocalories per gram. And so when we have fatty acids in the bloodstream, they're unable to pass into the cell due to the cell membrane. So in order to get across this, we use a fatty acid transporter. This brings the acid inside the cell. But this isn't the site of beta oxidation. Beta oxidation occurs in the mitochondrial matrix. And in order to transport the fatty acid into the matrix, 2 steps have to undergo first. So the first one is the coenzyme A will be attached to the fatty acid, and this is done through fatty acid acyl synthetase. And so here's the mechanism for it, and this should look very similar to an acetyl-CoA molecule, with the exception that the chain is much longer. So the next step will be the replacement of the coenzyme A with carnitine. So here's carnitine up here, and this is done with enzyme CPT1, which is carnitine palmityl transferase 1. And so now we have acylcarnitine. And acylcarnitine can actually be transported into the matrix via the CAT antiport, which is chloroamphenicol acetyltransferase. And so what this does is it exchanges one acylcarnitine from in the cytoplasm to inside the mitochondrial matrix, and while doing that it also will take one carnitine from inside the matrix and put it into the cytosol. So it'll go over, and then once inside the matrix, the acylcarnitine carnitine will be removed with CPT2, and we will be left with acyl-CoA, which is now able to enter into the beta oxidation cycle, but as well carnitine comes off, which allows us to bring in another acylcarnitine.
Thank you, Jason. So now that the fatty acid is ready for beta oxidation, this is a full cycle of beta oxidation, which is consisting of 4 steps. First step being dehydration, second step being hydration, then oxidation, and then dialysis. And so one cycle of beta oxidation produces one FADH2, which is flavin adenine dinucleotide, and one NADH, which is nicotinamide adenine dinucleotide. 2 cycles of this is enough to produce 16 ATP, which goes through the same pathway that we talked about in previous lectures for ATP synthase. And to understand ATP or beta oxidation further, the first step of beta oxidation is dehydration, which is oxidized by the enzyme acyl-CoA dehydrogenase, which removes the hydrogen group from the second and third carbon to produce a double bond. FAD is also used to produce FADH2 in this first step. And the second step of beta oxidation is hydration, which is a simple hydration mechanism between C2 and C3 and adds an OH2C3. This step is catalyzed by enol-CoA hydratase. Now, after we produce L-beta hydroxyacyl-CoA, it then goes into step 3, which Lewis is here to take over.
In the third step, which is the oxidation step, the hydroxyl group of the C2 of L-beta hydroxyacyl-CoA is oxidized by NAD+. So the enzyme that's responsible for doing this is 3-hydroxyacyl-CoA dehydrogenase, and the ending product would be the 3-keto-acyl-CoA. That being said, after getting the ending product, we go into the process of dialysis, which cleaves the thiol. The enzyme that's responsible for this is beta-ketodiolase, and the products, there's 2 products, acyl-CoA and acetyl-CoA. The enzyme that catalyzes this is, of course, it takes place between C2 and C3. Therefore, the end products are an acetyl-CoA molecule with the original 2 first carbons, C1 and C2, and an acyl-CoA chain, 2 carbons shorter than the original acyl-CoA chain that enter the beta oxidation cycle. So within this process, we really have to consider the cis and trans conformation of the fatty acids. So these are unsaturated fats, and the unsaturated fats bypass the first step and go straight to the second. So it bypasses this and goes to this. But it can only go to the second step if the fat is trans. So most fatty acids are in the cis conformation, which is unusable in beta oxidation. Thus, we need to transform the unusable cis form into that of a usable trans form. So change this cis into trans. So like what enzyme is responsible for this transformation, we'll go through the process in the future through our presentation.